right. Thank you so much for coming out on this rainy Friday afternoon. I apologize about the weather. In December, I'll do everything in my power to make sure we have dry skies for that Strand Training Day. So we are very excited to welcome Dr. Merabet. He's joining us today from Harvard Medical School, Department of Ophthalmology. He is active in clinical research relating to individuals' adaptations to blindness, so very, very applicable to the work we do here at MSD. As director of the Laboratory for Visual Neuroplasticity, Dr. Merabet's main research interests include the development of assistive technology for the blind and understanding how the brain adapts to the loss of sight, both from ocular and brain-based causes. So we're very pleased and excited to have him out here today. Just a couple of housekeeping things. If you didn't sign in already, just make sure you put your name on a piece of paper somewhere before you leave today. And please make sure that if you have questions, I know a lot of us are going to have questions, if you could hold them to the end or till the break time, that way we can get through all the really important content that he has got planned for us today. So please put your hands together. Welcome, Dr. Merabet.
Good afternoon, everyone. How is the sound? I don't quite hear it on my side. Everything okay? Yeah? Fantastic. So good afternoon. Thank you, Claire, for the introduction. It is an absolute pleasure to be here. Unfortunately, the weather wasn't cooperating, so I haven't seen that much of the campus, but it looks like you guys have an absolutely gorgeous and beautiful facility. It's just really, really nice to see you. I guess this means I'll have to come back. Just a couple of things I'll mention, a quick housekeeping. As was mentioned, if it's all right to hold the big questions after the presentations. Some of you may be thinking about an individual you're working with or something along those lines, and I'm happy to have that conversation. But just to keep the flow going, I know a lot of you probably want to beat the traffic and get home after this session today. If we can hold the big sessions during the question period, but if you have a burning question like, I'm going too fast, or I can explain something too well, please stop me. I'll do my best to give you more details about that. All right. Similarly, for our colleagues and friends who are visually impaired, if you want another sort of take on the slides, a little bit more detail of what I was explaining, please feel free to tell me. For our hearing impaired friends, if I'm going too fast, or if you need more time to spell out, for example, some technical terms, please tell me as well. I'll tell you a little bit more about myself. As was mentioned, I'm a clinician scientist. I'm an eye doctor. I'm an optometrist. So I see patients in the low vision clinic. And in particular, I see patients with brain injury and those individuals who have their vision affected by brain injuries like stroke, trauma. I'm also a scientist. As was mentioned, I'm very interested in how the brain adapts to visual impairment, both of eye-based causes and also brain-based causes. And we'll talk more about that, obviously, in the second half. And then the last piece I would say is now as I sort of get older in my career, I try to get more and more involved with advocacy. So for example, I'm on the board at the Perkins School for the Blind. Next week, as some of you know, there's going to be a 2-day workshop at the National Institute of Health on CVI. I think that's going to be a watershed moment for the field. I'll talk a little bit more about that as well. And then finally, I'm a Patriots fan, so all of you in Ravens wear. Yeah, it's hostile territory. I can feel it already. Okay, we'll try to get past that. Okay, so to get started, here is my first slide. When I was an intern in neurology, I had an attending doctor. He used to always have this expression, and I never ever forgot it. He said, neurology is just like real estate. The 3 most important things are location, location, and location. What was he talking about? What he was referring to is that different parts of the brain do different things, right? And this is uniform in all of us. The back of the brain, as you know, is responsible for vision. There's parts of the brain responsible for touch, for hearing, for language, for thought, for balance. And it's this relationship between structure and function that really is the basis of neuroscience and neurology. That's to say, if I know where the brain damage is, I should be able to predict what type of clinical deficit or issues that patient has. And similarly, if I know what type of problems that patient is having, I should be able to predict where in the brain that damage is. So it's that structure and function relationship that's really, really important to keep in mind. And that's what really drives our thought process. So let's just take a look again. Bootcamp sort of one-on-one, I should say, in terms of brain anatomy. You all know that the brain has 2 large hemispheres, a left and a right hemisphere. Deep in the brain is what we refer to as subcortical structures, and in particular the thalamus. The thalamus is like the relay station of the brain, and we'll talk a little bit more in detail about that. And connecting the two hemispheres is something called the corpus callosum, which literally means big body. This is the highway that connects the information in between the two hemispheres. So those are the three main parts of the brain. The two hemispheres, deep in the brain, what we call the thalamus, or subcortical structures, and corpus callosum that connects the information or processing the information between the two sides. If we split the brain in the middle, we call this a coronal view because it kind of looks like I'm putting a crown on, right? There are some important distinctions as well. So the cortex is this overlying area of the brain, and we'll talk a little bit more about what the cortex does. This is essentially the processing part of the brain. Deep in the brain as well, as I mentioned, these subcortical structures like the thalamus. This is where information comes into the brain, right? It's like the lobby of a building. I'll get back to that analogy as well. And connections deep inside here, the ventricles, you know, in the middle there, that's where fluid is, right? That helps to kind of dampen shock in the brain as well. So that's useful. And the white matter is the connections in between the brain that allows the brain to talk to itself, right? So cortex, where all the calculations are being done. White matter, where all this information gets exchanged.
Please excuse the interruption, but all students have left campus. Campus is clear.
And deep in the brain is the thalamus where information comes in. So try to think of those three levels, and we're going to kind of break that down a little bit for you. Right? So, as I mentioned, here's this coronal view of the brain, the outer layer of the cortex, which we call the gray matter, the inner part there, which we call the white matter, that's where the information gets passed. Right? We have this distinction. And as I mentioned, information enters from the outside world into the thalamus, and then the thalamus sends it to the cortex for processing. The important thing, I knew that was going to happen. The important thing to keep in mind is that this is not a one-way street. Once information enters into the brain, it's sent back to the thalamus. And the brain is constantly processing information in this way. What comes in is computed, and that information goes back to influence the further information that's coming in. So it's a constant iterative process. This is true for vision, this is true for hearing, it's true for touch, it's true for all the senses. So it's important to kind of think that when the brain is working, it's always online. It's always taking information, trying to understand it, and then trying to compare it with information that's coming in. That's how it's broken down, and that's how it's wired, right? As I mentioned, all the senses go through the thalamus except one, and that's smell. And that has to do with some evolutionary reason, we believe. But all the senses go through the thalamus, as I said, get sent to the cortex, and then the cortex goes back to the thalamus to influence further information that's coming. So think of the brain as this constant wheel of information, where it's constantly taking in information, analyzing it, and then trying to refine what's coming in as well, influencing what's coming in as well. So this brings me to an analogy that I like to share, and hopefully this is useful for you. I think of the brain like a high-rise tower. The cortex is like the cubicles. These where all the specialists work and make all the calculations. Right? The white matter is like the elevators. This is where people go on different floors and can communicate with one another. The thalamus is like the lobby. This is where everything comes in, and this is where everybody leaves. Given this breakdown, you can understand why damage to one part of the brain leads to different effects than damage to another part of the brain. So if I have damage to the cortex, a stroke, for example, maybe the neighboring areas, the neighboring cubicles, can do that calculation, and I can recover some of that function. If I damage the white matter, the elevators are out, maybe there's another way that information can actually get there. Right? We talk about neuroplasticity. Neuroplasticity is very, very much a rewiring of the brain, trying to find another way so information can keep communicating. The thalamus, however, damage to the thalamus, that's really, really hard on the brain, right? And we know, for example, babies who are born premature, or if there was a stroke deep in the thalamus, that tends to be really, really difficult because no information is coming in and no information is getting out. So I think it's a useful way to kind of think about it. If you know where the damage is, you can start making predictions of what you think can recover cover and what could be more detrimental. All right. The brain and visual development. This is David Hewell and Torsten Wiesel, two individuals who I really consider as idols, to be perfectly honest with you. And they shared the Nobel Prize in the early 80s for essentially discovering the visual cortex. Understand that up until that point, when we thought about vision and understanding what vision is, we were pretty much just focused on the eye. We thought that if we understood everything about the eye, we'd understand how we see. What these two gentlemen did is they really opened the door to say that no, to understand how we see, not just about the eye, it's understanding the brain and how the brain processes. And because of this, they won the Nobel Prize. So they did a lot of work, a lot of groundbreaking work, I would say. But one of, I think, the most critical studies that they did is they reared kittens under very, very specific conditions. So they would rear these kittens with one eye closed, for example, and then the other eye closed, or perhaps both eyes closed. And they would look at the effect on the anatomy of the visual cortex, and the physiology of the visual cortex, to see how visual experience affects brain development. The reason why this was important is today, the way we treat amblyopia is a direct result of that basic neuroscience reasoning. That's why I do what I do. I think that if we understand the anatomy, the structure, and the function of the brain, we can come up with strategies to help various conditions. So I think it was a wonderful paradigm to demonstrate the importance of why understanding the brain can ultimately lead, hopefully, to education and rehabilitative strategies. So because of this timing issue, they realized that this happened very, very early in the kitten's development. We call it the critical or the sensitive period, which is the stage during which the brain is especially sensitive to environmental stimuli. And it's crucial for development, right? And you all know this. It's a lot easier to learn a language, a new language at 6 than it is at 60. It's not impossible, but it's a lot harder. And the reason why is because the brain is very, very sensitive or very, very receptive to outside information. So why not just have that for the rest of our lives, right? We should just be always learning what we do, but at the same time, the brain changes its organization or changes its strategy because it also wants to consolidate what it has as well. So in the early years, when a baby is developing, that's the time to get all that information, right? Later on in life, you want to consolidate, keep what you have, and learn the skills that you learned early on as well, master the skills. And that's kind of how the brain has to change the overall strategy. So in humans, that critical period tends to be within 2 to 7 years. That's not to say that we can't learn after, obviously we can. But that period between 2 and 7 is particularly critical. And that's why jumping to the case in terms of CVI, early diagnosis is extremely important because we need to know early on, is this a significant factor? And how can we adjust that child development accordingly? A lot of details on this chart here, but I want to just kind of share with you some important takeaways. It says that the basic structure and functional framework of the brain is in place by the 2nd year of life. So by the age of 2, that's it. You've got everything you're going to go with. After that, I'm sorry to say, it's kind of downhill, to be perfectly honest with you. So after the age of 2, the brain is essentially just reorganizing itself. It's fine-tuning all the wiring and the machinery that it has. So you can understand that a brain injury prior to the age of 2 is really critical. That same brain injury at the age of 20 or 80 isn't typically as detrimental because of this timing issue. So timing is everything, right? Let's break this down a little bit more for you, okay? What I have here on the y-axis is change. And I have all these lines which correspond to different parameters of the brain. Again, the details aren't important, but the takeaway message is this. The only thing that changes after the age of 2 are these white matter connections, the actual wiring of the brain. If you look at the other ones, gray matter, surface area, cortical thickness, they're all pretty flat. So after the age of 2, it's really just about keeping what you have and refining what you have. Prior to the age of 2, it's a numbers game. Let's try to get it all much machinery as much as possible. What else do you know about the age of 2? Those of you who have young kids. The terrible twos, right? Isn't it interesting that when that child is like, yeah, me, yeah, I'm so important, it tends to be around the time that the brain is switching strategies, right? And I don't think that's a coincidence. I think it has to do with the fact that the brain is fundamentally saying, okay, let's take everything we have Let's refine it. And that's where a lot of these big issues like identity and self and so on are taking the shape. So there is very, very much a relationship between this strategy and how the brain develops as well. The other thing to kind of think about, which I think is important, is this critical period, as I mentioned, between this period of 2 to 7, that's also very, very much corresponds to when these white matter connections are changing. But I also want to be firm in this idea that the critical period doesn't close at 7. It just starts fading. And as I mentioned, it's a lot harder to learn a language at 60 than it is 6. But what can we know, or what can we realize, is we have to be that much more intense in our strategy, that much more engaged. It's a lot harder, and that's because the brain is becoming more and more wired and set in its ways. It's hard to induce those changes, like it is, or I should say at 60, than it is at the age of 6. Last thing, a couple things to mention to drive this point home. What I'm showing you here is a nice simulation. This was done by Alice Wade, trying to demonstrate the clarity of vision early on in life compared to later in life. So you see the face of this young woman here. Newborn, 4 weeks old, to 8 weeks old, to 3 months old, to about 6 months old. You see it changing pretty dramatic in just 6 months. But from 6 months to adulthood, it's just basically fine-tuning, right? So the early stages is when it happens. That's really the business side of things. And then again, I think you can appreciate why early brain injury or early sensory deprivation is so crucial, right? But this is when a lot of these things get really bad. Last thing I'll mention on this slide is it's important to realize that different aspects of brain development happen at different times. The early stages of brain development tend to be more related to sensory functions, vision, right? Hearing, touch. Next tend to be things like motor and language. And then finally, things like higher cognition, memory, executive functions, et cetera, et cetera. And this is important because we know there are domino effects, they're not. If there is early sensory deprivation, it will more than likely have effects in terms of motor development, language development, and also executive function. The takeaway from here is that timing is everything. You really, really need to be careful and understand what's happening in these early stages, because that's when things are typically most critical. Last thing I'll share with you, the details aren't super important. What I'm showing you here is a series of visual features like orientation detection, motion, color perception, the analysis of the two eyes working together. Over time, infants, young, older, all the way to older adults. And the takeaway message here is that different aspects of vision also develop on their own timeline. So not only does vision develop on its own timeline compared to hearing, language, and haptic, aspects of vision also develop on their own timeline as well. So motion perception, color perception, that tends to be a little bit later. Spatial acuity and contrast sensitivity tends to be a little bit earlier. So it also depends within the visual system when things develop also within the system. 2 quotes that I really, really like and I want to kind of share with you is this one by Aristotle, who said, to see is to know what is where by looking. And I can't think of a more concise statement to explain what vision actually is. To know what is where by looking. Because most of you know there is a where system, there is a what system, and looking means moving your eyes. And that in a nutshell is what vision is all about. Another great one that I love, this is by Helen Keller. She said, the only thing worse than being blind is not having sight and no vision. The only thing worse than being blind is having sight and no vision. And I think the takeaway message from this is that just because you don't have good visual acuity doesn't mean you can't see in your mind. And that's an important takeaway, and we're also going to show you how neuroscience has figured that out. All right. One thing also that I think it's important to think about, and I use this slide a lot when I'm talking to parents and fellow clinicians, is when you're talking about the visual system in the human visual system, or even the any visual system for that matter, there is 2 components to think about, right? There is the eye and there is the brain. And I like using this analogy of a digital camera. You can think of the human visual system as a digital camera. There is an optical component, which is the lens. This is responsible for focusing and controlling how much light comes into the camera. And there is the body of the camera, which is responsible for processing the image once that image actually comes in, right? So if you hold the digital camera, you squeeze the shutter, after a very, very brief delay, if everything's working well, you should get a picture on the back of the camera that looks very, very much like the visual scene that you're looking at, right? The human visual system works very, very much the same way. The eye corresponds to the optical piece, right? The iris controls how much light is coming in and out, and the lens controls the focusing and the clarity of the vision, or the clarity of the image. The brain represents the cerebral part, the brain, I should say, represents the processing piece. That being said, you can understand why an eye-based visual impairment is fundamentally different than a brain-based visual impairment. Going back to my camera, if I drop the camera and I crack the lens, but the body is still good, I just have to put on a new lens and I can take good pictures. But if I drop the camera and the camera body is somehow damaged, I can put all sorts of different lenses on there. It's still not going to take the pictures. It's very much the same analogy in this situation. And that's why, just as a preamble to CVI, I think a lot of clinicians, my colleagues who've done it, I don't understand. Child has, the eye looks great, I looked in, everything's clear, and the optic nerve looks good, retina looks good. I don't understand why this child has all these issues. It's because we're not talking about the optical part, we're talking about the processing part. And just to drive that point a little bit further, I have the pleasure of working with these two individuals who have CVI, Tina and Kendall. And both of them, as I mentioned, have CVI due to early brain injury, and they're both photographers. And when you talk to them, they tell you, I like taking pictures because the camera takes place in my brain. It allows me to slow things down, to zoom in, to take videos, to play it a little bit slower. So the camera allows them to kind of take control of what's actually happening around them visually. And I'll share some videos with you as well in our second session as well. So hopefully that's also helpful. So again, really not probably something you're all very, very familiar with. Information leaves the eyes, enters the thalamus, as I mentioned specifically, what's called the lateral geniculate nucleus. Projection goes to the back of the brain, the visual cortex. Information also is crossing, right? So the right side of the visual world is processed in the left hemisphere. The left side of the visual world is processed in the right hemisphere, so it's crossed, right? And some important information to take away from overall, again, just from Vision 101 or Bootcamp 101, there's some important things to think about. The way that the brain actually processes information is kind of interesting. The first thing we notice is that when we're looking at a picture, as far as the brain is concerned, it's upside down, it's flipped, and it's interesting also that it spends a lot more processing power in the center of vision than it does the periphery. And the reason why is because the visual system cannot have perfect resolution all the time. It's not like a picture. It has to move the eyes to sample at fine detail and then stitch it all together. And again, I'll show you more pictures of how it does. The other aspect that's important, again, this is a great example of why knowing the neuroanatomy is helpful. If knowing that the right hemifield or the right side of the visual world is processed to the left, and the left hemifield, the left visual side is processed on the right, knowing what that patient is going through tells me where that damage is. So if the patient tells me I can't see very well in one eye, but I can see just fine in the other, I know that's in front of the eye. But if they tell me, for example, I close one eye, I can't see on my right, I close the other eye, I can't see on my right, I know that's the brain. That's the only way that can happen. And this happens all the time in clinic. I have patients who come to me and say, yeah, something wrong with my right eye. I can't see on my right side. Something going on here. It's not your right eye. It's actually your left brain. So knowing how the two eyes respond, or where that deficit is, and if they're similar in both eyes, tells me that this is a central problem. It's not a problem. Again, a great example of why the anatomy is important. All right. Just going through this to give you a little bit more of some background in terms of the anatomy, then we'll get into the processing, which I think is a little bit more interesting. So as you all know, the eye has the lens, the image is focused on the back of the eye, the optic nerve, which sends that information to the thalamus. The thalamus includes a whole bunch of what we call these relay stations. The most important one is the lateral geniculate nucleus. This is important for enhancing that image quality that comes in. And then sends these massive projections, what are called the optic radiations, to the back of the brain, the occipital visual cortex. Once that information enters the occipital visual cortex, what's really interesting is the brain breaks it down. So the color of that image, the motion of that image, the overall shape of that image are all separated and processed in different areas. So that's something that's maybe not very, very intuitive. The image enters the brain, and the first thing the brain does is deconstruct it and uses all these specialists, and all those cubicles that I talked about, who are just interested in one particular aspect of that image. And then together, the brain reassembles it in a way that allows you to have a the view of the world around you. Let's spend a little bit more time thinking about this. So this concept of feature specialization is an important one. So we know that the first earliest part of the visual cortex, what's called V1, is essentially a sketch of the visual world around you. V2 is for stereo, right? It's fusing the two eyes together. V3 is for the form of that object. V4 is the color of that object. V5 is the motion of that object. And we go on and on and on. There are places specifically responsible for faces. There are parts of the brain specifically responsible for the perception of a human body. Now again, this doesn't correspond to the location in the brain. I just want to show you that the brain deconstructs that visual image. And it's the connections between all of them that allows us to see. There are roughly 30 different cortical areas so far, and they're always growing, that are responsible for visual processing. And as I said, each one of these areas is responsible for one specific aspect of the visual system. And the brain has to put it all together. Let me show you a simplified view of the visual system, and that's this figure here. So what I'm showing you here is basically a circuit diagram, this very, very complex wire diagram with all the visual areas interacting for us to see. And just kind of put this into perspective, here is at the very bottom the retina, the thalamus, and primary visual cortex. Right? That's the eye to the thalamus to the primary visual cortex. But if I'm an eye doctor and I'm spending my time here, I'm not getting the whole picture, am I? Right? All this is what you need. All this extra machinery is what you need to understand in order to figure out how we see the world. So it gives you some perspective. And when you're studying vision and trying to understand visual impairment, you really need to spend a lot of time in these higher cortical areas and how this information is exchanged to have a better understanding. All right, last piece I'll mention, and we'll start going through a little bit more some examples. The last thing I want to mention, and I alluded to this earlier, is that once information enters the cortex, the important thing to realize is that it doesn't end there. I'm sorry, am I going too fast? Or?
A little louder, maybe.
A little bit louder? I don't know if I can get the volume a little bit louder.
Let me put it off in here again. Sorry, folks.
Is this better? Is that better? Yeah, I can even hear myself now. Excellent. Thank you.
One more.
A little bit more?
Let me just push it up a little bit.
Sure. Absolutely.
Thank you so much.
No problem at all. Yeah, again, please stop me if I'm going too fast or.
Is that any better, folks?
How was that?
Didn't help. Yeah. It was worse.
Next step is swallowing.
Let me see if I can adjust things in the back.
I don't think it's going to help. I've tried that. Yeah, and also I apologize because I'm kind of churning my head. Maybe is that a little bit better? We'll try to get the volume up a little bit more, okay? Good. So the other thing I wanted to mention with this is that when we think about the visual system, we think the eye, the thalamus, the cortex, and we're done. It's actually not true. I really want to drive home this point that once information enters the visual cortex, the cortex goes back and talks to the thalamus. So vision is really a loop of information, constantly changing and exchanging information. This is important, for example, for eye movements. Where the brain decides to look next is dependent on what the brain is processing and influencing where the eye should move. You get some examples of that. Also, the overall exchange of information between all these visual areas. I'm going to give you a demonstration about that. So it's important to think that there is this constant loop of information that's happening. And the brain is constantly recalibrating, analyzing, and adjusting on the fly. This is true not just for vision, it's true for all systems. So, the other thing to think about is we know that there is a division of labor. You're probably all familiar with this. If you look at the general organization of visual areas in the visual system, we know that from the back of the brain, primary visual cortex, into parietal cortex, and eventually the frontal cortex, we call this the where system. Where things are in space tends to be analyzed through what we call this dorsal strain. And we'll come back to this later on as well for CVI. In contrast, where things, or how I can identify an object, is handled in the what stream or the ventral stream. This is our visual repertoire. This is where we keep images, our data bank of images, like faces, houses, and so on. And it's interesting because clinically, we can actually separate these two streams. I do want to drive home this point that even though this is useful, this dorsal stream and ventral stream, it's important to realize that they don't operate independently. And there's constant exchange between the dorsal and ventral stream, right? So they are working in parallel, but at the same time are fully integrated. And again, we'll get back to this point in the case of CVI. So why do we know this exists? It actually comes from adult brain injuries. So for example, we know that damage to the ventral stream leads to what's referred to as agnosia. Agnosia means lack of knowledge, right? Knowledge comes from the word gnosis, right? So for example, I can show a cone to these patients who have damage in the temporal lobe, right? And they'll say, I have no idea what this is. But then if you ask them, what would you do with this? They'll start combing their hair. Why? Because their wear pathway is still intact. They still have the spatial aspects of the processing, but they don't have the ventral stream aspects that allows them to identify it. Right? The counterexample is damage to the parietal cortex, and this is referred to as neglect syndrome. They can identify objects, but they completely ignore one side of the visual world. So for example, if you ask them to draw this clock, they'll only draw one side of it. If you ask them to draw this house, they'll only draw one side of it. They'll also only shave one side of their face. They'll only eat one side of their plate. Right? So very, very different presentation. And it's this double dissociation, this disconnect between being able to identify an object and knowing what that object does in space, or understanding the spatial relationship of things in space, that allows us to understand this dual divisional labor. Let me give you an interesting clinical example to kind of drive this point home. This is a classic case of neglect. This is a patient who came in the late 90s who had damage to his right parietal cortex. So he has neglect. What the patient was asked to do was to draw a cube. He wasn't able to do it. He was asked to draw a house. He can only draw the right side. He neglects the left side. If you give him a cancellation pass, where you just simply have to cross the lines out, he only crosses them on the right. He doesn't cross them on the left. Right? A couple of years later, he's readmitted to the hospital, and they retest his neglect. And you notice here, he's able to draw the cube, he's able to draw the house, and he doesn't miss anything on the cancellation pass. Anybody want to guess what happened? That would be a good in terms of our discussion. You're right. You would think it was rewired, but it's actually not what happened, because this essentially happened overnight. It was too fast for it to be rewired, but that's a good guess. I didn't tell you that piece. He was admitted to the hospital, like I said. It was an acute event, something sudden. In the other hemisphere, which balanced it all out. Right? Now, I'm not suggesting anybody has neglect. You should pray to them to have an episode of hemisphere. But it goes to show you that sometimes in neurology, less is more, right? And the brain is constantly trying to balance information all the time. And this very sort of unique case, a stroke in one hemisphere led to a deficit, then a stroke in the other hemisphere negated that deficit to begin with. So again, interesting relationship between structure and function. All right, let's talk a little bit more again from the visual development standpoint. Here's an important thing to keep in mind. It turns out that the ventral stream and the dorsal stream do not develop at the same time. Remember how I talked about different aspects of vision also develop at different timelines? It's also true for the dorsal and ventral stream. It turns out that the ventral stream develops first, earlier than the dorsal stream. That also means that the dorsal stream tends to be more susceptible to early brain damage as well, because it's still getting wired, it's still getting kind of put together. You're probably all familiar with this idea of dorsal stream dysfunction in CVI. That's part of the story. We think that the dorsal stream is more vulnerable to injury than the ventral stream. We think that's why in CVI, one of the main things that we see is this dorsal stream dysfunction. It is also important to get back to this idea of feedback. So from the eye to the thalamus and then back to the cortex, as I mentioned, and then over time. So motion perception is a great example. Very often I have teachers or colleagues of mine telling me, you know what, I don't understand. When this baby was really, really young, they used to follow all sorts of targets. And then after, it just wouldn't stop following anymore. I don't understand. Motion was really easy at first, but now it became really, really complicated. The reason why is because the brain is trying to use its cortex. In the early days, in the early years, it's essentially a thalamic system. It's just basically rudimentarily following motion. But because the brain isn't developing correctly, these feedback connections from motion processing areas back to the thalamus, that same complex motion is a lot harder for that individual to follow. So it's a difference between being able to say, yeah, that moved, and being able to say, yeah, this is my laser pointer. It takes a cortex to do that extra level of processing. So that's what may help explain the disconnect of why early on in early stages, the baby maybe orients very, very well. But then after that, you seem to not track and follow. Because you need that higher order processing to do that more sophisticated process. All right. Vision is an active process. We do not notice or process most of the visual information that enters our eyes. There are limits to the processing capacity of the visual system, believe it or not. And the eyes do not provide a high resolution or fine detailed information across the entire field of view. So when I'm looking, for example, here, my eyes are constantly moving around, taking snapshots, and my brain is stitching it all together. Now you may ask, well, why is that? Why is the brain dedicating so much processing power to the center? Well, if I had that same processing power for the entire visual field, my visual cortex would be this big. And you can't do that. So it's much more efficient to process only the center and use the eyes to move them around and then create and stitch the image together. All right? We'll spend a little more time thinking about that. The other solution, or I should say, the solution is to move the eyes. Right? So as I said, at any given time, a picture looks very, very focused, or the visual world looks very, very focused. But at any given time, the eye is constantly darting and sampling information about the visual world. The other thing that's interesting is that the eyes essentially don't see in between each saccade, each movement, right? That's a period where the brain is essentially shut off. The reason why is because it doesn't want to be seeing and have the impression that the world is moving, right? So the brain is only actively processing when the eyes are still. The brain is constantly taking all this information and then trying to stitch it. So if you have a child, for example, who's moving their eyes a lot more, you can imagine that's a lot more processing power on the part of the brain. We'll get back to why I think that's relevant in the case of CVI. Right. So over time, the brain stitches together a high-resolution representation of the visual scene, as I mentioned. The eye movement patterns reflect the content and the saliency of the visual scene, which I'll show you here. This was a classic work done by a gentleman named Jarvis. What he did is recorded eye movements using a very old contraption. Unfortunately, we don't do that anymore. And what he asked these participants was to just simply look at a face, and he recorded their eye movements. Now, eye movements are very, very jittery. They're very, very busy, but they're not random. And what you notice is the brain picks where we should look. We tend to focus on the eyes, we tend to focus on the nose, the mouth, the overall perimetry of the face as well. So the brain is constantly picking where we should look, where we think most of the information is, right? And that's all due to those feedback projections that I was telling you about. So the brain is deciding where the eyes should move to. Let me give you another example of how this can be a little bit complicated. So take a look at this visual illusion here. I'll describe it for you. If you move your eyes around, you'll notice these black dots, right, every once in a while. You'll also notice that you can't see all the black dots at the same time, even though they're always there. I'm not doing anything, right? I don't know about you, but this drives me crazy because I know the dots are there, but I can't see them all at the same time. This is your processing gone wrong. This is a visual illusion that tries to exploit the fact that as your eyes move, you are going to lose resolution in different areas. So this is basically what you don't want to see. I think it's a great example of how the eyes and the brain have to work together coherently in order to understand an entire field. A little bit more about eye movements and visual search, because this will become relevant to our conversation about CVI in the second section. It's important to realize that eye movements, again, tell us something about how you are analyzing the visual world. Where you look, what you don't look at, how long you look at something, tells me something about how you are processing that visual information or the visual world around you. So let me give you another example. This is again Jarvis, this early study. What he did is he showed a picture of this gentleman walking into a room with a group of other individuals. And he asked individuals to just freely explore the image. He didn't give any instructions. He said, just take a look at this picture. I'm going to record your eye movements. And what you notice here is the eye movements, where do they go? They tend to be on the faces of the individuals. They tend to be on the object, the picture on the wall, objects on the table. Right away, without any instructions, right? Then he told the same people, I want you to tell me how wealthy, or what is the material wealth you think of these individuals? And notice how the eye patterns now change. Instead of looking at the faces, you start looking at what the people are wearing. Certainly at that time, how you dress was a reflection of your overall material wealth. So same picture, very, very different eye gaze patterns. So where do you look, where you don't look, how long you look, tells me something about what you're trying to get at in the processing of the visual scene. Here's a clinical example. This was a video taken of two individuals talking to one another. So somebody is observing two people talk. And you'll notice that the overwhelming majority of the gazes tend to be on the face of one individual and the other face of the individual, and then going back and forth. Here's an individual with autism spectrum disorder. You notice they're not looking at the faces. They're looking at the hands, the body, all sorts of looking in the middle. So they're not picking up on the same cues as someone with neurotypical development. This tells me something, trying to relate any type of cognitive or developmental deficits, and their eyes tell me what may be related. They're not picking up on the same cues as someone with neurotypical development. Large takeaway from this, or the important thing to take away from this, is visual search or eye movements requires a lot of brain power, a lot of machinery. The eyes have to work together. Deep in the thalamus, there are structures that coordinate the eyes moving together. The visual cortex, parietal cortex, temporal cortex, frontal cortex, which is important for attention. So in the context of brain injury, either early in life or later in life, you can imagine that's probably going to manifest itself in terms of eye movements. So eye movements, in particular visual search, tells me something about the integrity of the brain and the visual system. That's why I think it's very, very useful. And again, we'll get back to this in the conversation of CVI. So let's do a little exercise together. Right? I'm going to show you a quick scene. Your job is to find the blue circle in this visual scene. All right? Everybody ready? You're going to look for the blue scene. Just raise your hand very, very quickly. This is a speed thing. You tell me when you see it. Everybody ready? Here we go. Yeah, easy, right? There it is. No problem. Okay? Let's do another one together. Find the blue circle. Ready? Yeah, easy, right? No problem at all. Here's one more. Ready? Yeah, it's coming, it's coming, it's coming. Those of you who got it right away, did you realize that there were two? What happened? What happened? Why was this so much harder? Well, let's go back to our structure and function. When everything was the same except color, basically all you were using was your color area to figure this out, because everything else was equal. When I asked you to do it, excuse me, let me go back. When I asked you to do it by shape, right, the blue circle versus the X's, the blue X's, you were essentially just using your shape area. When I asked you to identify the blue circle amongst red circles, you were using your color area. There's only one cue that you needed in order to do this. When I have to do it by color and shape, those two areas have to talk to one another. That's integration. That takes time, right? So you can understand that when it comes to processing information in a complex visual world, lots of areas have to talk to one another. And you can also understand the damage to the brain is going to affect that processing speed as well. That's what I think CVI is. It's a system that is not fully integrated in processing all that information. So everything really hinges on brain connectivity, its ability to get all these areas to talk to one another. Here's another kind of interesting demonstration. This is what I call context is everything. All right? So what you see here is two gray circles. One on the left is on a dark background. The one on the right is on a light background. I don't think it's too difficult to convince yourself that the one on the left is lighter than the one on the right. Kind of looking backwards. Right? The one on the left looks lighter than the one on the right. Everybody see that? Here's the weird thing. They're identical. Right? Show it to you again. See, the one on the left looks lighter. The one on the right looks darker. If I take the background away, you're actually identical. Why? Because your brain is using contextual information to make sense of this visual scene. When things are on a dark background, it's important to make things look a little bit brighter. When things are on a bright background, it's important to make things look a little bit darker so that we can enhance that contrast and make things pop out and become more salient. So your brain is constantly computing. And this really drives home the point. You don't see a snapshot of the visual world. You see a reconstruction of the visual world. And again, any damage to the brain, especially early in life, is going to throw that off somehow, for sure. Here's another one. This is what's called a dual or a bistable visual illusion. Some of you probably have seen this movie, right? If you look at this visual illusion, there are two options. You'll see either an old woman by her profile, or you'll see a young woman turning away from you. Does everybody see that? Yeah. It's interesting that you can't see both at the same time, right? You see one or the other. We call this a bistable illusion. Everybody get that? All right. I'm going to go one better. Who sees the young woman right away, predominantly? Yeah. Who sees the old woman predominantly? Less than. You're not going to like this, but it turns out what you see is actually correlated to age. The older you are, the more likely you're going to see the old woman. I'm not pointing anybody out. I'm just telling you that, right? So like I said, context is everything. In the outside world and in your head too. Another one. You all know FedEx? You all see these FedEx trucks? This isn't an ad for FedEx. Just trying to drive a point, right? Do you guys ever notice that there's an arrow in the logo? Yep. Yeah? Right? I'll show it to you if you've never seen it. It's right there. But that's not even the most interesting thing. If I take the arrow away, now you see it. Now, every time you see a FedEx truck, you're going to see it. I'm going to go one better. You're going to think I'm the one who told you that there was an error there. Exactly, and that's the point. This is called cognitive priming. So again, context is everything, and this is how we remember things, right? I can prime you to see things in the visual world, and you can remember the context of that information as well. I think that's a learning strategy. I think that's something that we should be leveraging. So the visual system and the brain are very, very responsive to these sort of things. One more, all right? I'm going to show you a picture, right? And here's your job. I'm going to show you a picture of a messy bathroom. And when I show the picture, I want you to tell me if you find the toothbrush. It's a speed game. So as soon as you find the toothbrush, raise your hand. Everybody ready? Okay? Here we go. Where's the toothbrush? Oh, easy. Right there. Right? How many of you saw this one? Look at the size of it. Right? How did you miss it? Right? So to show you, acuity is not everything. Right? So let's break this down from a neuroscience perspective. When I said find the toothbrush, this is what I think happened. Right? Your visual cortex opened up its filing cabinet. Your dorsal, or I should say your ventral stream, started thinking about what does a toothbrush look like? The texture, the color, the shape, etc. All the iterations that you have. Your dorsal stream started thinking, well, where is normally a toothbrush? Next to the sink, in my hand, in the water, in the cup. And you kind of had this sort of fuzzy image in your head, and as soon as I showed you, you went straight to it. That's priming, right? Why did you miss the big one? Because it's not in your repertoire. Right? But now it is. Because now when I show you that picture, you'll see it right away. Now it becomes obvious. It just goes to show you, acuity is not everything, right? It's really context. And that, I think, is a very, very important point that we should leverage. All right, a couple of last comments here, and then we'll take a break. So beyond the dorsal and ventral streams, I think this concept of dorsal and ventral streams is very, very useful. It's a great way to sort of conceptualize the brain, how it works. There are other streams that people are thinking about. There's something now we're talking about the lateral stream or the lateral pathway, which goes to a different part of the brain responsible for socialization and so on. I think the take home here is that we shouldn't be bound to one particular framework versus another. I think it's a useful way to conceptualize things, but I really think we should think about the brain more holistically. Because it takes all these parts working together in unison, well connected, for us to understand what's going on. So this was a piece that I wrote with John Ravenscroft in Scotland, thinking about the specific case of CVI, how we should go beyond just dorsal stream dysfunction, ventral stream dysfunction, think more holistically. And my hope is that different neuroscience techniques will help us get to that, and that's what I'm going to share with you in the second half. Last thing I'll just mention, which is kind of a strategy, is to think less about dorsal, ventral stream, or this part of the area, or this part of the brain, or like that, and think about it more as processes. Think about it as a bottom-up process. So for example, if I'm looking at this cat, what is particularly salient about this cat, right? The edges, the ears, the tail, the legs, the stripes. These are all what's referred to as bottom-up processing. Color, contrast, edges, simple things about the visual scene or the visual object I'm looking at. We call this bottom-up processing, right? In contrast, what I know about cats is what's referred to as top-down influence. So for example, if you are looking for your cat and you walk into your living room, you don't look on the ceiling, because you know that that's, I don't know your cat, but typically, cats are normally on somewhere on the ground or a piece of furniture. So your brain says if you want to find your cat, that's where you should look first, right? So that's experience-based. We call that top-down processing. The reality is that top-down processing is much more powerful. Well, that's talking about killing my whole story there. This is the battery. I am plugged in, believe it or not. I disconnected something. Sorry about that. Let me just do a quick. Yeah, that was good. Beautiful, thank you. That would explain a lot. Top-down processing, there's my big arrow, is experience. You know, you have expectations about how the visual world works. I can also drive that, as I showed you in my previous examples. So top-down processing is much bigger than bottom-up processing, and it's the connection between the two that matters. What's coming in versus what I expect to see should jive. And when they don't jive, then I get all sorts of funny things like those visual illusions that I showed you, right? In my mind, to cut to the chase, CVI is a condition where that top-down influence is not as developed because the visual impairment does not allow the child to develop that robust repertoire that normally you would expect for a child of that age. And on top of that, it creates a mismatch. So what's coming in versus what they expect to see doesn't connect. It's a different framework, it's a different way of thinking about what CVI is. Okay, I believe it still says it's running low, believe it or not. Now? I think so. It might be just, yeah, it just seems to be really sensitive. Okay, I promise I won't touch it. There we go. A couple of last comments before we wrap up this section. We'll get to a quick break and we'll move to CVI. So vision, as I mentioned, is an active process, right? So how much information can we handle, right? It's really easy to find a toy by itself. A lot harder to find it. Yeah, this one is this guy. It's just not cooperating. Excuse me, let me just make a quick change here. Hopefully that's a good sound. Good sound. A lot harder to find that same toy when it's in a toy box, right? Easy to find a person walking by themselves, a lot harder to find them in a crowd, same person, right? So the moral of the story is that there are limits to our abilities, right? When things are going well, this is not what's happening with my computer. This is so strange. It won't stay powered. Yeah, I think I have a break with that. I'm sorry about this. I really don't know what's going on. So when things are normally working well, right, this isn't a problem. On my computer.
Not work from the computer either.
Yeah, no, I can do it manually. So I'm just going to speak from this part and then we'll figure it out during the break. What I suspect is happening in the case of CVI is those reserves are not as robust. Right? Our ability to handle this complex information in our visual world is less in the case of CVI. This has to do with this early brain injury. We're going to drive home this point a little bit more in the second section. Loading capacity, again, just very, very quickly, it's important to realize that if we have a task that we need to do, and that task uses half of our reserves, right, we still have room to work with. But if that task takes all of our capacity, well, I'm going to have a complete shift. I wasn't far from the end, so this would actually be a good time to take a break. Let's figure this out. Okay, stretch our legs a little bit, and we're still plenty good for the rest of the day. So I apologize. Let's try to figure this out.
Break time, folks.
All right. So again, I apologize for the technical issues. It's funny. The first thing that popped in my head is like, somebody hates Tom Brady. I'm sure you guys. Absolutely. The one where I had the circles? Absolutely. All right. So some important takeaways from this, because I think it's actually related to the issue of rehabilitation and education as well. I think it's important to think about how task demanding something is, and also how much reserve and how much capacity you have as well. So let's just go through this to kind of drive this point home. So what I'm showing you here on the left is your ability or your capacity to do a certain task, right? That's your reserves, if you will. The task, which is in green, is about half full, which means I still have all this extra capacity to do more stuff. If the task gets harder, I still have reserves that allow me to do that task. If the same capacity gets filled, if the task is that much more demanding, then I don't have any reserves left, right? And what I think is challenging, particularly in the case of CVI, is that same task, which was not demanding initially, is now completely demanding because the capacity to begin it was limited. That's the point I was trying to make with this slide. So let me just sort of reiterate this. When it comes to getting better at something, there's two strategies. Make the task easier, or build reserves, build capacity. Right? That's the takeaway from this slide. They're not mutually exclusive. The hope is to do both. So simplifying the task, but also at the same time building capacity, building reserve, is how you can get through this kind of move. Does that make sense? Yeah? Okay. A couple of quick slides, and we'll switch to the CVI conversation. Hope to finish in about a half hour. The concept of neuroplasticity, which was brought up earlier today. What is neuroplasticity? Big fancy word we hear all the time. Neuroplasticity comes from the Greek word plastikos, which is where we get the word plastic from. And just like a piece of plastic can be shaped and molded into a different structure and a different function, so too can the brain. So plasticity refers to the ability of the brain to change its structure and its function in response to the environment, experience, even injury or sensory changes as well. That's the takeaway from it. It is not a guaranteed fix. Sometimes when I'm in clinic and I'm dealing with a patient with a brain injury, the family will say, okay, when's the plasticity going to kick in? When is this all going to just go away? It doesn't work that way. Plasticity is happening all the time. It's not a good or a bad thing, it's a fact of life. And the secret is to try to harness it in a way that we can steer the brain structural and functional changes in a way that's beneficial for that patient, okay? Here's a great example of what I mean by this. This is actually a picture that hangs in my office. It was taken in the late 40s, after the Second World War. And this photographer went around Europe, went to areas and cities that were shelled from the Second World War. And he found this young boy who was blind. And as you might imagine, this boy reads Braille, right? As you might expect. But there's more to this story. Unfortunately, one day, the same boy was out in a field, and he picked up something he shouldn't have picked up. It ended up being a landmine. The landmine exploded, destroyed both his hands, and he still reads Braille. Not because he sees the dots, but he's learned to read Braille using the tip of his nose and the surface of his lips, which is what you see in this next picture here. That is neuroplasticity. That's what I'm trying to get at. Make the task easier, build capacity. Right? That's the strategy when it comes to neuroplasticity. And there are a lot of great stories like this that give us this hope, this idea that, yeah, the brain can still change. With the right conditions, the right motivations, the right circumstances, we can create this change. So here's an interesting question I'm going to leave with you, and I think that leads into our conversation about CVI. As you all know, the back of the brain here is the visual part, the visual processing part. What do you think that part of the brain does if somebody is born blind? What does all this machinery that I've been blabbing about for the past 45 minutes do? Does it stay silent? Does it do something else? We've actually answered that question using functional imaging. Functional imaging is a way to see the brain in action. MRI scanner or PET scanner in this particular case. And what you see here is an individual who is blind, but their visual cortex, as you see in this circle here, is active. So why is this visual cortex active in an individual who's never seen? You probably know the answer already, because they're reading Braille. Right? It's also true that if you are born blind, you have a lot more activation than if you learn to read Braille later in life or lost your sight later in life as well. So there it goes back to that timing machine, right? Change can happen, but if it's going to happen, typically it's better to do it as early as possible. This is true not only for Braille, it's true for hearing, it's true for smell, it's true for even memory. So the take-home message here is that in the case of ocular blindness, the visual cortex, the part of the brain normally responsible for vision, gets functionally recruited to process non-visual information. Mother Nature does not waste brain cells, is the takeaway message from this. So now let's take it to the next level. What if you're born and your eyes are fine, but it's your visual cortex that's damaged? And that's the case of CVI, and that's why I think it's fundamentally different. And this is not a trivial issue, because I'm sure many of you now know that strategies that were developed for ocular blind kids are not necessarily working for kids with CVI. There's some overlap, but they don't take to it the same way. And I think the fundamental issue is because their brains are wired differently. I think that's why the neuroscience perspective of trying to look at ocular blindness versus brain-based visual impairment is important. And by trying to understand how the brain is fundamentally different, can maybe help us come up with some strategies. In the interest of time, I'm just going to go through here just to remind you that you can be born with an eye disease. You can also get an acquired eye disease later in life. You can also get a brain injury later in life, like a stroke. And you can, of course, have a congenital brain-based visual impairment as well, which is CVI. The important takeaway here is why do we distinguish brain-based impairment early in life versus later in life? And it has to do with loss versus lack. An adult who has a stroke, a brain injury, knows what's missing. They have something to compare it to typically. In the case of a child, that's just the way they are. That's how they've always seen the world. So the strategy that we typically develop for adult rehabilitation, in the case of acquired injury, may not be necessarily ideal for an individual who's always seen the world this way. Which is an important thing to think about as well. So hopefully what you've taken away from this morning is context and timing is everything. And if you think along those lines, things I think start making sense in terms of why there are so many differences. And the last thing I want to mention about this before we go on to specifically the CVI is it's important to realize that in adults, the overwhelming majority of causes of blindness and visual impairment is eye-based, macular degeneration, glaucoma, diabetes. In kids, it's brain-based. That's a fundamental shift in our thinking. At the Perkins School for the Blind, where I happen to be on a board, I work with a lot of collaborative studies, the majority of the kids are diagnosed with CVI, with a brain-based visual impairment. It's not a small population by any means, and I'm sure it's going to be quite similar here in the Maryland school as well. That represents a shift in our thinking and how we're approaching it. And my hope is that by understanding the brain a little bit more, we'll have a better understanding of what could be the next series of strategies. Okay, so let me just switch gears here, specifically to CVI. Computer, the battery seems to be cooperating, which is good. Now specifically to CVI. All right. This is the Perkins School for the Blind. Many of you know that it's the first and oldest school for the blind in the United States. It's a beautiful campus. It's in Watertown. You can see Boston in the background there. It's a few minutes outside. Helen Keller was a Perkins graduate student. It is really a fantastic institution with a long, long history. And this is actually where I was an intern. When I was an optometry student working with special populations and low vision, I actually interned here. And that was a big, big influence, as you might imagine, in terms of my work and my interest in working with the visually impaired and also with CVI. The reason I got involved with CVI was really a response to my friends and colleagues here at Perkins. They were saying, we have all these kids who are visually impaired. Some of them have 20/20 vision, but they're clearly visually impaired. Why are they fundamentally different than all those kids that we had, decades ago? And it's true. If you look at the typical Perkins child 50, 60 years ago, you find kids who are blind, profoundly blind, with some ocular disease or pathology or something. Now these kids are coming, and I'm sure it's similar here in Maryland, with a much more, let's say, complex profile, right? They have not only visual issues, but they have hearing, motor issues, cognition, memory, language. What's going on? Well, that has to be somehow related to the nature of that brain injury that's affecting many, many aspects of that child's development. And in many cases, these children have a visual acuity that doesn't allow them to qualify based on state statutory definitions of blindness. But they're clearly visually impaired. And I think that's an important thing to think about. This is a nice feature that was published by the National Eye Institute not too long ago. And it says here, vision loss in children with eyesight, whose eyesight may be 20/20, requires new diagnostic and teaching strategies. So you're like, how can you use 20/20 in vision loss in the same sentence? And the answer is there's a lot more to visual impairment than just visual acuity, as you all know. So I think it's an exciting time. This is Dr. Michael Chang. He is the new director of the National Eye Institute. Not too long ago, he presented what's referred to as the new plan moving forward for research by the National Eye Institute. Some of you may actually know this. There was a request for information many years ago, and I'm sure many of you actually wrote in as well. And 2/3 of the response that they got were CVI related. Think of all of the things that the NEI normally thinks about: eye disease, diabetes, infections, and so on. 2/3 of the respondents were CVI related, and this really got their attention. So I think now we're in a position where I think there could be monumental change coming. In fact, next week, tomorrow I fly back to Boston, and I got to come back next week and do this all over again. There's going to be the Trans NIH CVI workshop. It's going to be a 2-day event where we're going to try to tackle some of these difficult issues like diagnostics, definitions, and so on. I certainly would recommend to log on to the second day. You can remote in. I think it's going to be a very, very interesting conversation that I think could be pivotal for our kids with CVI. Just some comments here very quickly, again, in the interest of time. You all know that CVI is not only the most common cause of pediatric and visual impairment in the United States and in the developed world, it's also the most common cause of diagnosis now in schools for the blind in the United States. At Perkins, as I mentioned, it's more than half diagnosed with CVI. In a study that was done by CVI Now, which is a center at Perkins, they're doing some epidemiology work. You can see that it's quite staggering throughout the country as well. I think a lot of this has to do with resources, the ability of a family to make sure the child gets diagnosed. So I think the numbers are really, really underestimated. Here's a great study that was done in the UK, and what they found, or what they estimate, is that it's about 1 in 30 children are suspected or affected with CVI. That's one kid in every classroom. That's a big, big number. That's not a trivial thing by any means. And the question is where they're coming from. So as you all know, many of these kids are born premature, have some sort of perinatal injury, and it turns out we're getting really good at saving them. We weren't so good 50, 60 years ago, but we're good now. They're thriving, they're living long. longer, but we need strategies to take care of them, educate them, and help them live independent and full lives, right? Prematurity, for example, worldwide is a big, big issue. And you can see on this map here, the United States is a hot spot for prematurity as well. So this is not something I think is going to go away anytime soon. So, cerebral or cortical visual impairment, and after the question period, if you want to get back into the why cortical or cerebral and so on, we can certainly talk about that. But the functional definition that seems to be getting a lot of traction now is by Saki and colleagues. It says CVI is verifiable visual dysfunction which cannot be attributed to disorders of the anterior visual pathways or any potentially co-occurring ocular impairment. The take-home message is this: these kids are visually impaired largely because of a brain-based issue. They may have eye-related issues. They may have strabismus, they may have an eye chair or something. But the eye-related problems cannot explain everything that child is going through. That's the important takeaway. It's the brain-based issue, which is the bulk, or the bulk explanation of what the child's issues are. A couple of things to think about. Visual acuity can range from normal to profound blindness. There are some kids who are 10 to 20, and there's some kids who are profoundly blind. But the majority of these kids have some level of visual function, for sure. That's an important distinction. Some have visual field deficits, usually in the inferior visual field, and reduced contrast, and they also have ocular motor issues as well. But the important thing here is that an impairment in visual acuity or visual field is not necessarily required for a diagnosis of CVI. And that's important because if you live in a state that requires that definition, you need to have worse than 2200 or worse than 20 degrees of visual field, how do you get that child registered? In the state of Massachusetts, we've changed the language. That third level is, does that individual essentially function as a profoundly blind individual under certain circumstances? And that's what allows individuals who don't qualify based upon acuity and visual field to still be registered with the Mass Commission for the Blind. So it's that functionality I think makes the difference beyond just acuity and visual field. The second thing I want to think about is that obviously, as you know, the medical history is very, very complex. When I have a clinician or a colleague or a family member say, I think this individual has CVI, the first question I ask is, tell me about the birth. What went on? Were they born premature? Was there any maternal health issues? Any complications during the pregnancy? Anything happened right after? A trauma, infection, a seizure? And if there's something that happened around that time, that certainly raises the suspicion that that child may have CVI or some sort of neurological injury. That paired, obviously, with functional visual deficits. The other thing to keep in mind is a lot of the times, neuroradiology, that's to say brain scanning, will show you a lot of signs as well, some overall structural changes, but it's not in every child. There's some estimates now that I'm hearing out of Europe that say about 30% of the kids who are diagnosed with CVI have an MRI that looks normal. So you can't just depend on an MRI to make the diagnosis. It can be helpful, but it's not an actual requirement to make the diagnosis. It all depends on the function. All right, great study by Lisa Fazzi in Italy, one of really the pioneers in this area. What she did is she took a large group of patients, all with a brain-based or early brain-based injury, and she did a systematic evaluation of all these visual functions. So she looked at refractive error, in other words, did they need glasses? They looked at whether or not the back of the eye was intact, normal or not normal. Visual acuity, did they have an eye churn, impaired contrast? Did they have, for example, visual field deficit and so on? She went systematically in all these kids and found that a lot of them had these signs. The interesting takeaway from this is the overall majority, or the most common feature, is that these kids had visual perceptual processing difficulties. That was the most common feature in all these kids. Whether or not they had reduced acuity or not, they all, or the most majority, had perceptual problems, the way that they perceived the visual world. The way that they used it or assessed it is something called the developmental test for visual perception. It's a neuropsych test that we use. Some strengths and weaknesses with that, and I'll get back to that with time. But just to show you that it's the perceptual piece that's important to think about. It's also useful, when we talk about CVI, to distinguish these visual function aspects, like acuity, contrast, visual field, and these functional vision aspects, like perception, how they actually perceive and interact with the visual world as well. So I want to be a little bit clearer about that. So what do these kids say, typically? We hear things like, I have a hard time finding my parents in a crowd. Or if you give me, for example, my favorite toy, I can identify it, but if you put it in the toy box, I can't find it anymore. That's not visual acuity, that's processing. That's the brain trying to figure out what is dealing with that information that's coming in. And Gordon Dutton uses a term what's referred to as dorsal stream dysfunction, because what he's found is that on the net, overall, a lot of these kids have visual spatial processing issues, which tends to be described with the dorsal stream, as you all know. Right? So where does this all come from? How can we assess this? This is Dr. Barry Cram from the Perkins School. What we know is that when we want to make a diagnosis of CVI, there's lots of things we have to think about. As I mentioned, the history is extremely important. The medical history really, really drives the suspicion. But we also have to be creative. We can't just rely on an eye chart. Right? You need other ways to try to, for lack of a better term, reveal or expose the visual deficits that child might have, right? So a lot of these kids, for example, they can even read the eye chart, or maybe 20, 30, 20, 40 visual acuity. But if that doesn't explain why they can't find their way through a crowd, or can't cross the street safely. That's clearly the processing piece of the brain that we need to try to understand. All right? And just a comment here, there's a lot of gurus, or a lot of publishing, I should say, trying to drive home this idea that we need a multidisciplinary approach. At the Perkins School, Dr. Cran works very closely with TBIs to get a sense of what are these kids like in the classroom? What are these kids like at home? What are their parents telling us? My personal opinion, the parents are the most informed individuals in the room. They tell you what their kids are going through, and that has to be folded into the diagnosis as well. There's only so much we can do in 15, 20, 30 minutes. But where that child is in the real world tells us a lot more about what's going on. So just want to distinguish visual function versus functional vision. Visual function are things like acuity, contrast, visual field, basic aspects of how the visual system works. Functional vision is how you use your vision in the real world. Driving a car, in a classroom, finding your favorite toy, finding a sibling in the crowd. When things are working well, typically these two go together. But in the case of brain-based injury, particularly in the case of CVI, they're not always related. So a child could have very, very good visual function, but very, very poor functional vision. And why is there a disconnect? What we try to do from a research standpoint is answer that question using a variety of techniques, which I'll share with you today. Again, I want to be cognizant of time. So we developed novel tasks using psychophysics to allow us to look at different aspects of vision, not just acuity. We also use virtual reality and eye tracking, which I'll show you as well, to create environments that are similar to what that child goes through and see how do they interact with these virtual environments. And the third piece is using what's referred to as wearable eye tracking technology. So here, you see this individual here is just doing a simple task of picking up these blocks and matching them. That red dot corresponds to where they were looking. And we can use that to extract their eye tracking data, doing real world tasks, for example, we do in the classroom. And that's what I want to share with you. And the reason, as I mentioned, that we think this works is because there is a connection between where you look and how the brain is trying to process that information as well. I just want to drive home this point here is that there is no one lab or one approach that's going to cover this all. CVI is an extremely broad and heterogeneous population, so there's not going to be one test or one approach that's going to cover everybody, right? We really need a collaborative effort to do this. I just wanted to show you here that these are 3 labs. That's us on the right. This is Dr. Melinda Chang in the middle at UCLA. This is not UCLA, the University of Southern California. This is Glenn Prusky from Cornell Medical School. Glenn Prusky tends to work in the NICU, babies, right away, getting a sense, can they fix, can they follow? Can they give me something about the integrity of the brain? Melinda Chang tends to use eye tracking for basic tests where they're nonverbal. They can't, for example, repeat letters and so on, but their eye tracking, for example, on a teller acuity card tells me what they're looking at. In our group, as I mentioned, using virtual reality, we tend to work with, quote, older and, quote, higher functioning individuals, adolescents, who are verbal, who can actually describe their visual impairments and challenges as well. So the working hypothesis is this: the eye and the brain go together. And we think that the visual system of an individual with CVI is more sensitive to increasing visual task demands, environmental complexity, and may rely on different visual cues compared to neurotypical visual development, right? So how they interact with the world, what they extract from a visual information standpoint is fundamentally different than what we typically see in neurotypical development, all right? We have done focus groups We brought in kids with CVI, families, TVIs, to try to help us understand what some of the challenges are. One of our first studies that we did was developing virtual reality environments, and we learned, for example, children have a hard time finding their favorite toy in their toy box, for example. Or they may have a hard time finding a sibling or a loved one or a friend in a crowd. So we use these sort of information from focus group studies to create virtual reality environments, to try to recreate these scenarios and see how these kids actually interact. So well beyond what is typically done in an eye exam. So let me show you the first task. This is called the virtual toy box. The task is very, very simple. You have to find this toy. They choose which toy they want. In this case, it's a blue truck. On the left is a control, and on the right is an individual with CVI, the same age. And all they have to do is just find the blue truck when it appears, okay? And just make a saccade and fixate on it. I want you to compare the movement of the control versus the right on CVI. This is what it looks like. You see how the control goes straight to the blue truck, whereas the CVI individual takes a much more complicated path and gets there? Show you a couple more examples. Here's a color match. This is when two toys have the same color. See how the CVI individual has to go back and forth, right? They get there. If you just looked at the start and you just looked at the end, you'd think that the kid with CVI did just fine. But they're clearly working harder, right? And a lot of our kids with CVI tell us that after a day of coursework, I'm exhausted, right? Why? Because they're working so much harder to get to that point. So for example, if I'm walking from here to the back of the room and I take a direct path, I'll get there. But if I also take it, for example, a much more circuitous pathway, I'll also get there. But at the end of the day, I'll get a lot more tired. And we think that this has a lot to do with what's going on here. Their eyes are working much, much harder to try to extract that information. Now, I know what you're thinking. You're probably saying, well, how do you know this child maybe just doesn't have an eye problem to begin with? Well, that's actually not the answer. Here are the same two individuals, left and right. Left is the control, right is the CVI. And when the toy is by itself, look how the CVI child is able to get right to it. So this is not a problem of the eyes being able to move. This is a problem of the environment, right? This is the task that's driving these differences. So we've looked at the data, and indeed there are indeed differences. So what we do is we do this toy box task, as I mentioned, and there are 3 levels of difficulty, low, medium, and high. And what changes is the number of unique toys in the toy box. And we suspect that their performance should get worse as the task gets harder. That goes back to this idea of load and difficulty and so on. And this is indeed what we find. If you plot success rates in controls who are matched by age to the CVI individuals, overall, the CVI kids do worse. They miss the target more often than our controls do. The other thing to keep in mind is look at that variance. Look how much more variable it is in CVI compared to controls. Controls tend to all perform about the same level, but our CVI group is much, much broader in terms of how they're performing as well, which again speaks to the variance in the overall heterogeneity of CVI as well. The other thing that's interesting now is if you separate those CVI kids based on their visual acuity, so the orange bar in the middle are kids who have 20/20 or 20/25 visual acuity, essentially normal, versus all the kids in the right bar, in the red bar, who have reduced visual acuity, 20/30, 20/60, and so on. They do as poorly. Right? So visual acuity, good visual acuity, does not make this go away. This is a processing issue. This is not an acuity issue. Last thing I'll mention on this slide here, if you plot reaction time, how fast they find the target as a function of task difficulty. So the higher you go up the y-axis, the slower you are. The more you move to the right, the harder the task is. In blue, this is what it looks like in controls. Pretty flat. As the task gets harder, their reaction time is pretty much the same. This is what it looks like in CVI. There's a baseline that's higher, they're slower, and the other thing that you'll notice is they're more sensitive to load. As the task gets harder, they get slower, certainly in a more dramatic way than we see in controls. So CVI is a condition where they can be very, very sensitive to task demands. This is crucial from a clinical standpoint, because if I have to just read these letters on a chart in my eye exam room, that's nowhere as hard as it is crossing the street. So there's a disconnect between what we measure in the clinic and what these kids are really going through in the real world. Last piece of evidence I'm going to show you here is you might say, well, how much of this is actually perceptual? And how much of this is really maybe attention deficits, right? How much of this is attention? Well, it's a hard thing, it's a hard question to answer. But what we did is we studied a group of kids with ADHD. Sorry, is the data not there? Oh, I'm sorry, I don't have an updated slide. We did the exact same study, with kids with ADHD. So these are kids who have a diagnosis of attention deficits, who aren't born with a brain injury, and their deficit looks much, much closer, their profile looks much, much closer to what we see in controls. So we know that this is more than just attention, is what I'm trying to say. Another task that we did for our older participants is looking at whether or not you could find an individual in a busy hallway. In this particular task, all you have to do is find the principal of the school, and we have them by gender and race matched. You pick the individual you want to look for. On the top right is an example. The control is on the left, the individual with CVI is on the right. And you can see the gaze pattern, they lock on for the control, but there's a delay for the CVI participants to lock on as well. So when we add this motion and this complexity, we see again these deficits appear. And very, very similar to what we saw in the case of the toy box, overall, our CVI kids do worse than our controls, and there's a lot of variance in terms of their performance. Similarly, what we saw as a function of the response time, there's that load effect that we see in CVI, not as pronounced as what we see in the case of controls. And here's that piece of information I told you about. This is what it looks like in ADHD. They look like controls at the low level, and then as the task gets harder, they do worse, but it's nowhere similar to the profile that we see in the case of CVI. So attention has a role to play, but we think the bigger aspect is the perceptual aspect as well. Okay, I'm just trying to keep cognizant of time here. I want to just cut to the chase if I can, in terms of where we're heading next, just so we can wrap up here. One area that we're starting to develop now, going beyond simple eye tracking and virtual reality, is to track their eyes in the real world. And we do this using what's called wearable eye tracking technology. These are glasses that you wear. I can also put in your prescription if you need glasses to correct as well. And then the eye tracking follows your eyes in the real world. That's my nephew, by the way. He'll do anything for candy. That's how I was able to get this picture. And when we're developing the technology, he was our first subject. So I'm going to show you here in this video what the data actually looks like. So this is just me. You'll see that in a second here, I'm going to hold up the card. That bing is the calibration. That's it. I'm now collecting data. This is me looking inside the lab. Here I'm reading an eye chart. I'm just a typical Snellen eye chart. You'll see me looking off in the distance. Here I am looking up close with an iPad, and that red circle is where I'm looking, overlaid on top of what I'm looking at. This is me jaywalking in Boston, probably not a good idea. But again, it works outside, works indoors as well. And here's that task I was telling you about. Even like a visual motor task, something simple like that, I can use the eye tracking. And that red circle corresponds to where I'm fixating at any given time. The larger the circle, the more time I'm spending. Once you extract that information, the software can recreate where you were looking. Right? It's a registration of what you were looking at the video, and what I do is I take a snapshot of the visual scene, and it does a one-to-one mapping and recreates the visual scene. So, when I was looking at that eye chart, it creates a heat map. It tells me exactly where I was looking at. I can also recreate a sequence of gaze points as well, one by one, a sequence of all my gazes. So I can extract the data from the real-world perception into tasks that I think would be relevant for the case of a child. And you can use this in the classroom. We're thinking about now doing a study where you look at cluttered classrooms versus non-cluttered classrooms and how that child stays focused, for example, whether or not these changes can change their overall ability and how they perceive the visual world. And a recent area that we started working with, which I'll share with you and I'll end on, is using eye tracking during neuropsych testing. So when a child does a neuropsych test, like the developmental test or the BERI or something like that, and the child does poorly, you really don't know why. Was it a motor issue? Was it an acuity issue? Was it a cognitive issue? Was it the ability to use the marker and so on? So we think eye tracking can allow us to disentangle how much of it is largely visual, how much of it is motor, and maybe what is the integration between the two. And that's what we've been playing with that now. So here's an individual with CVI doing the developmental test for visual perception. That red dot is his eyes, and in the upper right is a heat map that's being generated. So you can see that's his gaze as he's doing the test. And that heat map is being generated online. I know where he's looking at any given time doing the task. What's interesting is if you now look at the data, here it is in a control. So these are all the targets. These are the drawings below, targets, These are the drawings below. Here's an individual with CVI who did quite well, and you can see they're getting progressively more and more fixation as the task gets harder. Here's an individual with CVI that didn't do as well. Look how that gaze pattern is a lot more, more staggered, more sort of random. We can put numbers to this. What we're finding is our controls in blue spend a lot more time looking at the drawings than they do the target. And our CVI kids are the opposite. They spend a lot more time looking at the drawing than they do studying the target. Sorry, I shouldn't say actual drawing. So it's two different strategies. How CVI kids are doing this is different than kids with neurotypical development. I think that's the important takeaway from this. I'm going to skip this video here again in the interest of time. Couple more things. This is the cancellation test. You probably know this one. Just goes to show you that we can actually look at left versus right and see whether or not there's an attentional deficit. We are now doing visual motor tasks as well. This is a task that allows, this is an individual with CVI. The task is simply to take the blocks and then place them. And look at the strategies here. You see, for example, he's going to use his index finger to see he feels it with his finger before he puts it down. Right? You wouldn't get this if you just looked at the starting point to the end point. And by looking at his eyes and his hand, we can separate how the hand is moving versus how the eyes are moving and give us a better understanding. Is this largely a visual perceptual issue or is this a motor issue? Last thing I want to share with you, just out of curiosity, why I think the eye tracking is useful and compelling. I went to some colleagues at Perkins and I said, just give me one of the worksheets that you have in the classroom. And this was a simple task here. It was called a word search. You have a whole bunch of pictures like this. You have a bunch of words here. You got to cross it off and then find out where the object is. Who thinks this is a good idea for kids with CVI? No, definitely not. Way too cluttered, way too much going on, way too condensed. I'm going to show you how the eye tracking actually demonstrates this. Here's a control individual who did the task correctly in 1 minute. Here's an age-matched individual with CVI doing the same task. A lot more work. They did it correctly. They got it all right. Took a lot more time. But does this make sense to put a child through all that work? Maybe they're better served, again, doing something else with that time, right? So how we design the materials is extremely important. And as I said, they are extremely sensitive to load. They are extremely sensitive to the complexity of the visual scene. And I think eye tracking can kind of get at that. But then we can also look at interventions. If we simplify things, does their eye tracking behavior change as well? So again, I want to be respectful for time. I have other things. It's really up to you, Gar. I know people want to do traffic. I'm happy to stay and keep the conversation going. It's really up to you.
Maybe we can do, does anybody have any burning questions that the whole group might like to hear?
Absolutely. I may have other sort of slides as well. Again, I want to be respectful of time. I apologize that things took a little bit longer than long. I have a
I see a
question.
couple hands. I'm coming with the microphone, okay?
So when you were doing those eye tracking tests and the virtual worlds, how did you accommodate for novelty and complexity, all those things did you pre-teach? So how did you take those factors out to know what exactly was the effect in which area?
Absolutely. So there's a couple of things that we did with those designs. Take the toy box, for example. So the first thing is they had to choose the toy, right? We didn't force any particular toy on them. They had a choice of 3, and those for 2 reasons. One, we want them to be kind of engaged in the task and have sort of sense of control, and control that novelty, if you will. The second is we needed to know that they could identify the object by itself. Right? So that was the first level. After that, the environment stays the same. We only manipulate the task difficulty. So we were very, very careful to try to only, so there's a great saying in science research, we say make one change at a time. If you make multiple changes and you see a difference, it's very, very hard to understand what caused that difference. So in that particular case, we only changed the number of unique toys in there. The set size was always the same. It was always the same number of toys. So we looked at, for example, color. We looked at the background. We would make one change that would allow us to associate behavior based on the one change that we made. So that's a study design issue.
Okay, folks, I do want to be respectful of time. It is almost at the end of our day. So Dr. Merabet has very kindly offered to stay and answer any questions if you do have additional questions. However, we know that some folks got to clock out. You have things to do. It's Friday, so if you need to go, let's give a quick round of applause. Dr. Merabet, thank you so much for coming out. If you have